Hello everyone, we are here to learn about the next topic, that is bacterial reproduction or bacterial recombination. That is, in short, the reproduction methods of bacteria. So as we know that there are three methods of reproduction of bacteria. First one is conjugation in bacteria. Second one is the transformation in bacteria, which is given by the scientist Griffith. Third one is transduction in bacteria. So conjugation, transformation and transduction, these are the three methods of bacterial reproduction. Out of that, we discuss here the third method, that is transduction method in bacteria, which is also called as an lytic and lysogenic cycle. So in case of transduction, there are two cycles are involved, that is lytic and lysogenic cycle. So we discuss here what is lytic and lysogenic cycle, and togetherly it is called as a transduction in bacteria. Okay, so first we discuss about the bacteriophage means what. Before going to that transduction topic, you should know that what is meant by bacteriophage or the structure of bacteria or structure of that bacteriophage. So first we discuss about the bacteriophage is nothing but the virus that infect the bacteria. In one word definition, which is compulsory, ask for your midterm. What is meant by bacteriophage? Phage means virus particle or virus, and bacteria means bacteria. When virus kills the bacteria or when virus control the bacteria, is called as bacteriophage, or simply it is also called as an bacteria eater. Or bacteriophage means what? Virus eating bacteria. That is virus that attack on the bacteria and they kills the tissue which is present inside the bacteria or the material which is present in bacteria. So this is called as an bacteriophage. Then next we discuss about the bacteriophage, the structure of bacteriophage, that is virus. As we know that the outer coat of that virus is called as an capsid head, or simply it is called as an capsid. So this is the first outer side, that is red colored indicated, that is capsid head. Second one is, as we know that the structure which is present inside the virus, inside the capsid head, that is called as an DNA. So this is nucleic acid, or it is called as an DNA is also present inside that, that is called as an DNA. Double stranded DNA is present inside the virus. Third one is the collar. Here the small circular portion just below that capsid head structure is present, called as an collar. Then whiskers, the thread like structure is attached to the collar, which is called as an whiskers. Then sheath, the spring like structure which is present here, it is called as an sheath of virus. Then at the bottom of virus, the plate is present or attached, this is called as an base plate, and on the base plate the tail fiber is attached, which is responsible for the attachment on the bacterial cell wall. Then spikes is also present at the bottom of the virus. So this is the structure of bacteriophage or the virus. Okay, the next we discuss about the next part. This is also the structure of bacteriophage, as we already discussed: capsid head, nucleic acid, collar region, sheath, base plate, spikes and tail fiber. So these are the part of or the structure of bacteriophage as we discussed earlier. Next we discuss about the steps which is involved in lytic and lysogenic cycle. That means what happens in lytic cycle and lysogenic cycle. So lytic means what? The breaking. When the virus particle enters inside the bacteria, so the breaking of bacterial cell takes place along with the multiplication of virus. How? So we can discuss here. So this is lytic cycle. So for that we discuss here the first steps which is involved in the lytic cycle, that is attachment, second one is digestion, third one is injection, fourth one taking control, fifth one that is multiplication, and sixth one is rupturing or breaking. So let us see one by one the structure. The first step in a lytic cycle, that is the attachment. In this step, the bacteriophage, that is virus, that attach itself by the tail or with the help of tail to the cell wall of bacterium. So as we know that this is the cell wall of that bacterium, and on that cell wall of bacterium the virus get attached with the help of tail fiber. So very simple steps is there. So what is first step? Virus, they comes in contact with the cell wall of that bacteria and they attach with the help of the tail fiber. So that's why it is called as the attachment. Okay. Second one is digestion. So can we discuss, or we can discuss also here the diagrammatic representation. Here the virus, as we know that first step is virus, they comes in contact with the virus first comes in contact with the cell wall of the bacteria. As we know that this is the cell or the bacterial cell. This is the bacterial cell. Here the bacterial cell, and that virus, they comes in contact with the, this is virus, they comes in contact with the bacterial cell which containing the bacterial chromosome. Here it is bacterial chromosome which is present inside the bacteria. So first attachment, that is virus or bacteriophage, they comes in contact with the cell wall of bacteria, first one. Then next we discuss the second one, the second step, digestion. After the attachment, then next one is digestion. So in this step what happens? The bacteriophage or virus which contains an enzymes called as an lysosomes or lysoenzymes which digest the cell wall of bacterium, and thus the opening of bacterial cell wall is formed in the bacterial cell. That means what happens here? So what happens when the bacteriophage or virus, they comes in contact with the cell wall of bacteria? So as we know that virus contain lysoenzymes. So that lysoenzyme, they acts on the cell wall of bacteria. Due to that, the opening of cell wall of bacteria is takes place, or the cell wall of bacteria is opens. Okay. So this is about the digestion, that is the digestion of cell wall or opening of cell wall of bacteria is takes place in second step, that is called as an digestion. Okay. And third step, as we discuss here, the third step after digestion, third step is called as an injection. So in injection, the bacteriophage, that is virus, it contracts and inject its DNA through the opening. That is, whatever the bacterial cell is open, so through the opening, that as we know that the virus contain DNA, so that DNA is enters inside the bacterial cell through the opening inside the bacterium, while the protein coat and the tail remains outside. Here, as we discuss that in second step, here we discuss about the lytic cycle. So what happens in second step? The phage DNA, phage means what? Virus DNA, viral DNA. Whatever the red colored viral DNA is present, so that red colored viral double stranded DNA, they enters inside the bacterial cell through the opening of bacterial cell wall here. So that is entry of the viral DNA is takes place inside the bacterial cell which containing only chromosome inside the bacteria. Okay. Then third step after the entry of virus inside the bacteria, the third step is taking the control. That means what? Once inside the bacterial cell, that bacteriophage DNA takes over the biosynthetic machinery of the host. That is, that bacteriophage or the virus, they take the control all over the bacterial cell, and at the same time that viral DNA that bacteriophage or that viral DNA replicates or it synthesizes its again the copies of own DNA and protein molecules. Here in fourth point, that is taking control. What happens once they enters inside the that viral DNA, once they enters inside the bacteria or bacterial cell, so what happens, they secrete, or that viral DNA secrete some enzymes, and due to that it the breaking of the chromosome or the bacterial DNA is takes place. Here the bacterial DNA, it's the breaking of bacterial DNA is takes place due to the secretion of enzymes by the viral DNA. That is red colored is viral DNA, isn't it? So what happens at the same time, when the breaking of that bacterial DNA takes place, at the same time what happens here, there is a replication of that viral DNA is also takes place. That is the new fake DNA forms using the nucleotides from the former host DNA. So there is a formation of new viral DNA or own copies of viral DNA is formed after the breaking of the chromosome or after the breaking of that bacterial DNA here. Next step after that taking control here, the multiplication. So what happens in that step, the bacteriophage multiplies and increase its number, hence form a lot of daughter bacteriophage. Thus the daughter bacteriophage exert pressure on the cell wall of bacterium. So what happens here, once they enters after the replication of viral DNA, that is red colored, and the bacterial DNA, what happens after the replication of both, what happens, there is a formation of, and in next step what happens, there is a formation of protein coat, and that viral DNA, and that viral DNA is directly transferred into that the protein coat. Here there is transfer of that viral DNA inside the protein coat, it takes place. That is the host cell transcribes and translate the fake DNA, producing the fake proteins. That means after the breaking of viral DNA and bacterial DNA, there is a formation of the protein coat, and due to the formation of protein coat, what happens, that viral DNA they transfer inside the protein coat, and there is a formation of new virus particles at the same time. So what happens, due to the formation of more and more virus particle or more and more bacteriophage inside the bacteria, then what happens, that bacteriophage exert the, or that virus particle exert the pressure here, exert the pressure over the wall of the bacterial cell, and due to the tremendous pressure which is exerted by the bacteriophage by the over the bacterial cell wall, there is a bursting or breaking of bacterial cell wall is takes place, and due to that rupturing of bacterial cell wall, what happens, the bacteriophage is released outside, and they again infect the new bacterial cell. So what happens, there is a breaking of cell wall of bacteria is takes place here. So this is called as a lytic cycle. So this is about the lytic cycle of the bacteriophage. So again we discuss here what happens. First one is the bacteriophage binds the bacterium, that is virus particle comes in contact with the bacterial cell, or that virus particle comes in contact with the cell wall of bacterial cell. That is first step, attachment. Second, what happens, digestion, that is the virus which contains the lysoenzymes, with the help of that lysoenzyme the digestion of cell wall of bacteria is takes place, and due to that the opening of cell wall of bacteria is takes place in first step here. The next one, what happens, due to the opening of or the disintegration of cell wall of bacteria, the viral DNA which is present inside the bacteriophage or virus, so that red colored viral DNA enters inside the bacterial cell. Then third one is the injection. Once the viral DNA enters inside the bacterial cell, So that viral DNA secrete or viral DNA take the control all over the that is taking control all over the bacterium. Once viral DNA enters inside the bacteria, so it secretes some enzymes, and due to that, what happens, due to the enzymes or nucleic acids which is comes inside the cell wall of bacteria, due to the nucleic acid there is a breaking of the bacterial DNA is takes place here. Next one, what happens, that at the same time when there is a breaking of where bacterial DNA takes place, at the same time there is a replication of DNA is also takes place, or new formation of new fake particles and formation of protein coat is also takes place at the same time. Once there is a formation of protein coat is takes place, so that viral or red colored DNA is transferred into the protein coat. Then there is a formation of the, once the protein coat is formed, there is a formation of the virus particles inside the bacterial cells, and more and due to the formation of more and more virus particles inside the bacterial cell, that virus particle exert the pressure over the cell wall of bacteria, and due to that the breaking of the cell wall of bacteria is takes place. So the new fake particle or virus particle is released outside, and again they again they control or again they infect the new cells. So this is about the lytic cells. Due to the breaking of cell wall is takes place here. The next, they comes in contact with, once the lytic cycle is completed, they comes in contact with the, or they enters in lysogenic cycle. So both are different, but the initial procedure or initial steps are same. So what is the initial step when they enters in lysogenic cycle? So the first one is, first the bacteriophage binds the bacterium. First in case of lysogenic cycle, the bacteriophage or virus particle comes in contact with the cell wall of bacteria, or attachment of virus particle to the bacterial cell wall is takes place, first one. Second one, digestion of bacterial cell wall is takes place due to the lysoenzymes, as we already discussed in lytic cycle. Due to the digestion, there is opening of cell wall of bacteria or bacterial cell wall is takes place. Due to the opening, the virus particle enters inside the bacterial cell wall. Once they enters, in case of lytic cycle, what happens, once the viral DNA enters inside the bacterial cell, so they get recombinant, or what happens, they get integrated into the bacterial chromosome. Whatever the bacterial blue colored chromosome is there, that viral DNA is integrated or recombined with the that chromosomes or bacterial chromosomes. Okay, so when they are recombined, so it is called as an prophage. It is called as an prophage. Then next, what happens, the next step is the multiplication or the reproduction of the bacterial cell is takes place. So what happens, that bacterial cell, as we know that this is your bacterial cell, that bacterial cell undergo binary fission. Binary fission means what? Cell division is takes place. Bacteria cell or bacteria get divides, and at the same time when the bacteria start to divides, so there is a formation of two copies of that viral DNA, and the chromosome or bacterial chromosome is takes place, and one copy is transferred into the daughter bacterial cell. There is a formation of constriction here, the formation of the constriction in between these two cells. Okay, so chromosomes, here it is written in fourth point, the chromosome with integrated prophage replicates, that is replication of that chromosome with prophage, that is replication of that viral DNA and chromosome is takes place by the formation of the constrictions here. The next one, what happens here in next step, again once the bacterial cell, after the replication of that prophage, what happens, that sometimes that viral DNA it gets separated from the chromosomes. In next step is the bacterial viral DNA or prophage is separated from the chromosome or bacterial chromosome. Okay, so here it is given in fifth point. In rare cases, the prophage may excise, that is, it is removed from the host chromosome, and the cell wall or cell will enters into the lytic cells. That cell is enters into the lytic cell. So this is about the lysogenic cycle. Here there is no breaking or lysis is takes place, no lysis of bacterial cell wall is takes place. So this is about the lysogenic cycle. Okay, so again we see, again once time again we see the lysogenic cycle. What happens first? In short, there is a attachment of virus particle to the bacterial cell wall, first point. Second one, the digestion of cell wall of bacteria. Due to the digestion of cell wall of bacteria, due to the lysoenzymes, that is lysoenzyme acts on the bacterial cell wall. Due to that, the opening or pore is formed, and through that opening what happens? There is an entry of viral DNA inside the bacterial cell is takes place. Third one, that viral DNA is integrate or reintegrate or recombine with the bacterial chromosome. It is called as a prophage. Then that prophage and the chromosome, bacterial chromosome and prophage replicate itself, and the daughter cell is formed by the formation of constriction or the bridge in between that two cells. Okay, after some time what happens here? Whatever the prophage in these two cells, that prophage it get excised or separate from the host chromosome, and the cell will enters into the again lytic cycle. So that kind of lytic cycle and lysogenic cycle that are takes place continuously. So lytic means what? The breaking or virus enters inside the cell wall of bacteria, and breaking of cell, a bacterial cell is takes place. And in case of lysogenic cycle, what happens? Virus enters into the bacterial cell, and only cell division is takes place with the prophage, and that prophage is excised from the chromosome. Only that procedure is takes place. So this is about the lytic and lysogenic cycle. Again, one explanation is also given here. You can refer that PPT also, how or what are the steps. I will send the PPT on your group. You can also see the red colored that lytic cycle and the blue colored lysogenic cycle. The simple step is given here, and this is for, you should also write the points which is written along with the image for your midterm examination, for your final semester end examination. So this is all about the point, or here point that we control. Again we discuss in very short how that process takes place. Here, as we know, that bacteriophage, that is virus particle. Again, for your information, again we discuss that same cycle. Okay, so this cycle, whatever the cycle or whatever the image we already studied, so same image will be there, but it is in the simple format it is given, so don't be confused. So this is bacteriophage, that is virus particle. This is phage DNA, that is viral DNA. This is host DNA, and here host bacterial cell. Okay, so what happens? As we already discussed, bacteriophage, they comes in contact with the host bacterial cell. Okay, that is attachment. Second, what happens here? Digestion of the cell wall is takes place by the enzyme lysoenzymes. Due to that, opening of bacterial cell wall is takes place. Okay, the next second step, entry of that phage DNA. That phage DNA enters inside the bacterial cell. Okay, and once that phage DNA enters inside the cell, so that phage DNA along with the nucleic acid, they enters inside the bacterial cell, and due to that nucleic acid, the breaking of the host DNA is takes place. At the same time, the replication of RNA, viral RNA or phage DNA is takes place, and at the same time there is formation of protein coat. Then next, what happens here? After the formation of protein coat, what happens? That viral DNA get or that phage DNA get transferred into the inside the a protein coat, and there is a formation of virus, a number of virus particles or number of daughter virus particles. Due to the extreme number of, or due to the multiplication of virus particles inside the bacterial cell, what happens, there is a, that virus particles exert the force or pressure over the cell wall of bacteria, and due to that what happens, there is a breaking of cell wall of bacteria is takes place, and due to that, that fake particles or virus particle get released, and this is called as an lytic cycle, that is breaking of bacterial cell is takes place. And when lytic cycle completed, they enters into the lysogenic cycles. So in case of lysogenic cycle, this is lysogenic cycle, the fake DNA infect a cell, same procedure, and they enters inside the cells. Second one, the recombinant, that is the fake or profake or the fake DNA becomes incorporated into the host cell or host DNA or genome. Next one, what happens, the cell divides, as we already seen, that is the binary fission, that is a method of reproduction of bacteria. The fission, that is breaking of cell is takes place, and during breaking, the replication of that host DNA and the viral DNA is takes place. The cell divides and profake DNA is passed on the daughter cells, that is, it is that daughter cells, host DNA or the fake DNA is transferred into the, or profake is transferred into the host DNA, and daughter cell is formed. Then what happens here, when it become mature, so under stressful condition, the profake DNA is excised from the bacterial chromosome and enters into the lytic cycles, just like this. Again here the lytic cycle is also given. What happens, that we already told. Okay, so whatever the points which is written just below the image, so you can draw the cycles and you should write the information which is given just below the diagram. So you can write as it is, there is no separate